presenting his work with microstegium that he did as part of the RU program, not this past summer, but the summer before. Nick is an undergraduate here at Eastern and an honors student who I've been fortunate enough to work with a little bit with running buffalo clover, or he helped out with some of my work this summer while he was there doing a photography project with EKU natural areas. He's involved with all kinds of research with Dr. Koslow, LP, Dr. Callie. I believe he's still wrapping up some stuff with Dr. Ruhfel, student body vice president, and I can go on. So without further ado, let's hear from Nick.
Great, thank you so much, Ted, for the kind words. I will get the screen shared. And how do I present slideshow? There we go. Okay, hi everyone, I hope you're doing well. So just to say the title, of course, Microstegium Migraine, Understanding Japanese Stiltgrass Ecology and Lily Cornet Woods. And I know some of the words might be foreign to a lot of people, but I'll break it all down. And this is for the BIOS seminars. I just want to preface with, this has been one of my goals since freshman year to present at the BIOS seminar. I went my freshman year and didn't understand those presentations at all. They all went over my head, especially the microbiology ones. It looked like alphabet soup on the screen, and I still don't understand some of them, but I've definitely grown as a biologist and a scientist, and I'm just extremely honored to be able to present at this seminar. And I know it's over Zoom, but the premise in itself is a huge honor. So to kick it off with an introduction, Microstegium viminium, that is the scientific name for Japanese stiltgrass. It is an invasive species now in the United States, originally from China. In 1919, it was introduced into Tennessee from porcelain packaging because it grows abundantly, so they can use it as support for the porcelain so it doesn't break. But has since, over the 100 years it's been in the United States, spread to much of the eastern United States, as far south as Florida, all the way up to some of those New England states, as far west as Oklahoma, all the way to the coast. So it is quite a nuisance, but it really becomes a nuisance because it can do this in the forest floor. This is just a complete covering of the forest floor. What makes Microstegium a deadly combo is it is a C4, meaning it usually does super well in huge sunny areas along with other grasses, but it's also shade tolerant, so it can grow in the middle of a densely covered forest. So those two combined make it a really powerful invasive species in the United States, which is not good for the nutrient cycling because it speeds up those processes. It also decreases the biodiversity. Specifically, it also decreases the biodiversity in lily cornet woods. The study site from Kevin Pacinda's research with Dr. Koslow the year before I did my research. So we were working in an environment we already had previous research, which adds to the validity of the argument to research it more. And then the study site is Lily Cornet Woods. It is in southeastern Kentucky. I've fallen in love with these woods. If nobody has been, I want to emphasize using that natural area. It is a special place. It changed probably the research I'm doing, and that's why I wanted to do it in there, mostly because it is old growth, which means it has not been clear cut or bulldozed, basically. So it's an intact forest, and it's a perfect base study system, which is super important when it comes to invasion ecology. Also, it's mixed mesophytic. This is a complex word described by Lucy Braun, and this basically means that it is rich in diversity, both overstory in the tree, so it's not like an oak hickory forest. It just has a myriad of woody specimens that dominate the forest ecosystem, including hemlock and beeches, buckeyes, and Ted can tell us all about the trees as his research he has done with those species. It is a very cool forest, but here's one of the beech trees that they have there. So the objectives of the study, just to put it plainly, we were hypothesizing whenever you go into that forest, you'll see that microstegium often occurs in canopy gaps. So we were thinking that there would be some correlation between microstegium and canopy gaps. So that was one of our hypotheses. And then our other one was we wanted to understand the behavior of microstegium over a larger scale in the United States. And we looked at the northeastern United States specifically, but we did some niche modeling with that. So some of the methods we took. First, we did plots. So we identified microstegium plots in the forest in the Shop Holler region, which is one of the hollers that they have there. We found 41 sites specifically. These were other ones that Kevin Facenda found, but these were also ones that I found. So we tried to identify every location of microstegium in that area of the forest. And then we used a coverage class when we visually identified them. And then this is important because you see that these aren't equal distribution or bins. That's because when you get into the 1% and 2%, telling the difference between those is very easy to our eyes. But when you get into the what's the difference between 50 and 49, it's really hard for us to be able to tell. So these bins make it easier for us to put them into categories. This is a modified brown blanket, so we're using Lucy Brown again in the study. And then for canopy openness, we decided to take a technological route. Nobody has used a hemispherical photography in their research of studying microstegium. They have used one of the concave densiometers that you hold in your hand, you look, and it points upward, which is of course a great method, but we wanted to use something a bit more current, which is just a different approach. And we did that with all the 41 sites. And then this is a picture it takes, but when you binarize it, which is an actual verb, is it turns the light into white pixels in the trees or the leaves, and the foliage and the closure into black pixels. So this has technically thousands and thousands of data points because each pixel is one data point for each picture. So we create a ratio and that gives us the percent open and closed the canopy is. And then lastly, we wanted to look at the biomass. So what I did was once I finished all the field measurements, I went about, picked up all the microstegium, patted off the dirt, and then dried it at 60 degrees Celsius. It was fun. I got tired of picking it after 14 sites. Our sites were 4 by 4 meters, so 16 square meters total. So it was quite a big area of microstegium I picked, but it was a really fun process to also just remove the microstegium from the forest. People have worked with it and researched it, but they still leave it after the years, which is ethically kind of hard to do. So Dr. Koslow and I talked and we wanted to get rid of it, which was, I think, a good decision overall. And then here's an example of those plots. So here on the left is pre-picking, and then on the right is post-picking. And you might notice I missed a few, but it got very tedious. But they do pull very easily. They don't leave too much dirt. And after you pat it off, I put it into a bag to take back to the university to dry. And I actually went back to the same exact site to where I picked, and there was a successful picking and removal of the microstegium from hand pulling. There are different methods you can take, but with isolated populations like this in the forest, I think a chemical agent would do more harm than good. So pulling it individually is just a better method, in my opinion, for these isolated populations in the middle of old growth forest, because we do want to remain that, keep that level of old growth and not try to tamper with it too much, of course. And then maxent. So maximum entropy is what maxent stands for. It's been around for around 2 decades, so not too long when you look at the grand scheme of things. However, there have been a lot of people that use it, and you can use it for modeling of rare species or invasive species or just common species, of course. But we did the invasive species, which requires a whole different set of methodology. And I thank Robert Pace specifically for helping me with this process. Without him, I definitely wouldn't have been able to accomplish the research when it comes to the maxent work. It is quite a robust program, to say the least. So the results and discussion, the good stuff. First, I want to talk about the field assessment, since those can kind of be broken down into 2 separate categories. So here's a map of Lily Cornet Woods. It looks like a seahorse to me always. But if you look at the triangle specifically, that's where we found the plots, and that's the Shop Holler where that, the short trail it's called. So we did, I don't know if people can see my mouse, but if you look at the left side of the short trail, which you can kind of see some of the deep incursions that microstegium makes. And you also might notice that it has that loop around where the topography starts to flatten out. That's where the gravel road is. So you can associate the spread of it to humans. And I would say, in my opinion, that it's definitely been spread by visitors, which is not optimal, of course. However, it does happen when you take people on these trails. It's made those deep incursions. But that's why we did some of the removal of those far-reaching plots. To talk about some of the data specifically, in the plots we had a average coverage of 25% when we did the coverage classes. In the canopy openness, which was kind of surprising, it was only 11% when you looked up. So that was kind of a surprising number, because you stand in the middle of a canopy gap and you're like, oh, this is probably 50%, but some of those only maxed out to 25% canopy openness, which was kind of surprising to me. But the camera doesn't lie. The camera's going to be better than my eyes. And then when it comes to biomass, I picked a lot of microstegium. This is the dried weight specifically. So I picked 0.6 kilos of microstegium, which was fun. And it took me a few weeks, but we had to do it in a short time because it grows. So we didn't want the biomass to shift too much, of course, over time. But each site had an average of 45 grams and total 631 grams. So the big thing we wanted to do with this is correlate these variables and see how much they were correlated with one another. So our original hypothesis was saying that if we have an canopy openness, the percent coverage would be highly correlated or somewhat correlated. And then the biomass would as well with the canopy openness. And we did that last percent coverage in biomass as a check on our methodologies. I'll explain that a bit later. So to kick it off, canopy openness and biomass, it was not correlated and it wasn't statistically significant, which was like, what? Like it went against our hypothesis and we were like certain, but I think when I really did a lot of reflecting on it, we saw where microstegium grew, but we didn't see where it did grow, which was some of those like shallower areas with high moisture levels and deep enclosures. So I think that, in my opinion, we were seeing, of course, it grew well in these canopy open or canopy gaps, but it did also grow well in non-canopy opens and just along creeks, for example. So this was an interesting graph between canopy openness and biomass. And then similarly, canopy openness and percent coverage with 41 data points was also not correlated and wasn't statistically significant. So we found that it disproved our hypothesis. However, upon further reflection, even though it's not statistically significant, it does have biological significance because this is basically saying the microstegium will grow anywhere and it doesn't care about the canopy, which makes it even more potent again to go along with its shade tolerance and its ability to make these deep incursions in our old growth forests. So even more of a priority to remove it and get rid of it in these ecosystems. However, to verify our methods, I did do a correlation between percent coverage, which is that visual estimate of how populated a site is of microstegium, and then biomass, the physical characteristics, and it was statistically significant at less than 0.005 of a p-value. And then also the adjusted R-squared value was 0.5471. So I don't know the crowd exactly, but for ecologists, this is a really exciting R-squared value. Having a R-squared of 0.5 is really good. So we did see that these two were correlated and it did go with just verifying our methods. Now into the MaxScent. So MaxScent has two components, basically, the GPS coordinates. This is an old map I made of the forest, but it just shows more brightly the coordinates specifically. And then we use those along with GPS coordinates from citizen science derived data points of microstegium. So GBIF and IDAG-Bio are both repositories of data, specifically occurrence points of microstegium, along with basically any other living organism, which is a really untapped resource, in my opinion, that should be incorporated in many studies. So I derived those, and we have 1,800 occurrences, basically, in our MaxScent model. So first what I did was, or actually, pardon, let me talk about the bioclimatic variables. The other part of a MaxScent model, along with the occurrence points, is the bioclimatic variables. These are for world climb. And again, thanks to Robert for processing a lot of this, but this is a lot of variables. And when you're running this model, it takes hours upon hours. So first, what we wanted to do is get a smaller list of which of these variables are actually being used in the model. So we ran a smaller model with less replicates to just get an idea. And then we narrowed it down to these 8 bioclimatic variables specifically so that we could just get a better idea for a more robust models with more replicates. And then after that, we put the settings in with the 1,800 GPS coordinates, 8 bioclimatic variables with 1,000 replicates. It total took 45 hours to run, thanks to the herbarium for using the computers there. And then total it generated 46 gigs of data. So it was about a gig an hour of data, which is astonishing for the number of, for just a model like that. It was really cool to see that. This was my first time really dealing with big data in this manner. So it was cool to just get this experience under my belt. So this is what it generated, the visual interpretation. And just a few things you notice, of course, in the Northeast, it's highly, highly suitable for microstegium growth. And then you are like, whoa, let's look at Tennessee and Kentucky for a sec. And you're like, okay, so not really that much. And which is very confusing because it was first introduced into Tennessee and then spread, of course, north. So it would take most time to get to those other areas. However, after further investigation and just plotting the points onto a map, we see that there are some biases when it comes to citizen science. So this is kind of one of the points of course, MaxNet models are great, but they're as good as the data, and the data is very biased along with population. And also the Northeast is just more active when it comes to being involved in citizen science and iNaturalist documentation of species. So I'm going to talk a bit later about how we're starting to address some of those problems in the MaxNet model, because it does have a very strong bias towards population. Even when you plot like a city like Boston, you see there a high amount of microcesium. And so does that mean that microcesium grows really well around cities? I would argue no. It grows where there's disturbance along roads and along these rich areas of a bank of water, for example. So it wouldn't grow in the middle of a city. However, it does because there are humans there, they're documenting it. So there's just an intense bias along with the model. So looking at the distribution, it is really cool to see that. And I was actually, when I gave this presentation, one of my friends in the REU, she actually lived in one of the counties that wasn't populated in green, but she said it grows everywhere there. So the MaxNet model can kind of point into areas where we should look for the species and where it's not documented in certain distribution maps, which is a really interesting output that you can get from a model. So the big picture, the really important parts of why do we do this? First is ecological investigation. What is the ecology of microcesium? That is super important whenever we want to move forward with removal and prevention. That's something that we should talk more about in invasion ecology is, of course, we want to remove species and prevent them, but we need to talk about like preparing our ecosystems so that they don't have a niche to fill in our forests. And then invasion behavior is just really important to understand of where will it grow? Why does it grow there? And that's coming back to the canopy openness investigation. And then again, why is this important? Biodiversity preservation of our forests. When you have not just microcesium, but the bigger picture of if we have 10, 20, 30, 40, 50 species that are invading our forests, that's just taking away niches for these native species, which again, we need to preserve for other parts of our wildlife pollinators as the plants are the foundational part of an ecosystem. And then again, I think that as humans, we're the cause. I think we need to constantly remind ourselves that the reason they're here is because of us. So we need to be stewards of the land and really make sure that we protect our lands as much as they protect us. So back to the back to the migraine. I titled this because I think in science, a lot of the times in these presentations, we talk about the good data, the good stuff, but I wanted to talk about the bad and what sucked about the research sometimes and the doors that kept closing in front of me, which is sometimes frustrating. So the model, that is probably the biggest part of my migraine, specifically the occurrence points. So, well, I'll talk about the bioclimatic variables first, but what we had to do was we wanted to find a better way, and I looked back in the literature of narrowing it down because when you narrow down the bioclimatic variables based on your biased occurrence points, then technically your variables are already biased. So what we wanted to do is we ran a correlation matrix among the 23 and then removed any that had a R value or a correlation of higher than, I think, 0.8 or 0.9. I can't remember exactly, but that's beside the point. We now have 5 bioclimatic variables, and we saw that when you look at these specifically, a lot of them are based off one another. So annual precipitation is one, and then another one of them is annual precipitation during wettest month or driest month. So they're based on one another. So we'd expect that they would be correlated, of course. And then occurrence deletion, that is been an interesting point. So when you zoom into the model specifically, each pixel is a 1 kilometer by 1 kilometer grid, basically. So what I did was I went to all these occurrence points, basically, and looked to see if there were 2 in one grid cell, and then removed them based on a random generator to pick it out so that we have less bias on density of growing. And then after that, we're going to create some new models, which I'm hoping to do in the coming week, which is super exciting, of course, to have a final model. And then, of course, writing has been a huge difficulty for myself with writing up something that is very confusing because MaxNet is based on statistics. So just communicating the results for MaxNet specifically has been quite a burden, but it is an interesting challenge, of course. And then the upside, of course. So the upside is we're going to be making new models, which is very exciting. I'm just really intrigued to see what they're going to show. Maybe less of a density up northeast and maybe more of a habitat suitability towards our state like Kentucky or Tennessee. And then writing, I'm very excited to get it written up finally and submitted. I think we're aiming for Castania right now, but we'll see wherever the piece takes us. So something else I wanted to talk about is the learning outcomes of research. And I know that the field is highly dense in faculty, so I wanted to really bring light to this. And I know that we're in a teaching institution, so we do pride ourselves in the delivery of certain classes, which is great. But I think that I wanted to really talk about the learning outcomes of research specifically, because we're not a research institution. However, we can use it as a mode to teach, or of course, and I can learn. So here's a list of the 20 that I wanted to talk about they literally have no overlap. I made it intently. Some of these I could break down. So like R, for example, I did coding for the data analysis, but also did model or the graphs. So both of those could have been broken down, but these are very general. So there's more than 20 in my opinion. Specifically wanted to talk about these niche modeling. This is just a super fascinating part of the research to me, and I've learned so much from it, really diving into the research and understanding what is going on up front and then behind the scenes, which is so confusing because the statistics in it are so robust and very few people even understand it. From what Robert and I have talked about, it is very confusing. And then collaborative research is super important, connecting with other people at the university and outside the university, getting their opinion on the research. Some of the steps we could take differently has been super interesting. Experimental design this was a very new concept for me. This was the very first experiment I literally designed on my own, hypothesizing from the original part of our research with Dr. Koslow all the way to the end where we're at the publishing stage, which is super cool. And again, publishing is a very weird concept. It is very complex, coming from the cost to publish to picking a journal, which one is best for the audience that you want to give your results to, and problem solving. Of course, that has been a huge learning outcome for me, because again, the migraine that I've gotten from this has just been it's been repeated over and over again. And then something I really wanted to stress is the combination of all. I would challenge somebody to find all 20 of these in one course at EKU, and that's what research can bring, is a combination of these 20 or more into one credit hour that I took with Dr. Koslow, and of course research over the summer funded by the NSF-REU program. And then big takeaways. I wanted to jump back out to the really big picture of what I learned from this, other than how to interpret the data. Citizen science is so important. I need to take better steps personally of uploading the pictures I take to iNaturalist. And right now, Dr. Watson is really beating me in that that count of uploading some occurrence data. But that is, if without the research, half of my research would not, without the citizen science, half of my research would not have been possible. And then something that was really jarring to me is educating the public. I think a lot of the times in science, there's that final step that is not taken of going from the super complex findings that you made, going back to the very simple, rudimentary takeaways from it. So I personally wrote a KNPS, or Kentucky Native Plant Society, article on how people in our organization can identify microstegium very easily with that silver line that runs down the midrib, or and then the best ways to remove it depending on how big your population of it is in your private land, because it's great if we remove it on public lands, but it's still going to be persisting on private lands. So there has to be both of those parts being attacked at. And then it just as a shameless plug, if anybody would like to write an article for the Lady Slipper, the newsletter for the Kentucky Native Plant Society, we're really wanting some writers. So that'd be a nice thing to add to your CV, of course. And then the biggest takeaway, I would say, is the internships, is really emphasizing that this internship did change my research path immensely. Without this, I would not be able to apply to these grad programs that I'm currently applying to. So I just want to emphasize that if there's a professor maybe that hasn't taken an intern on before, I really encourage you, because thanks to Dr. Koslow, Dr. Watson, Robert also helping me, and all the other professors that have helped me along the way, that this has immensely shaped my collegiate career. And then, of course, I'd like to thank all the people up here in the agencies that have funded it. Robert Pace with the MaxEnt modeling, Dr. Koslow, with many steps that I couldn't even list. Dr. Watson for the data for creating the maps along with advice and mental sanity help. And then Dr. Richter and Dr. Brown, who both were the program directors at the time, and then Curtis, who dealt with me and my just existence at Lilly Cornett Woods as he oversees that. But I thought during COVID times, it wouldn't be right if we didn't make sure that they were all socially distanced and masked up. Is I bet that they would be disappointed if I put them next to each other and not socially distanced. So thank you all for your help. And then with that, I would love to take any questions ranging from the research to the internship in itself.
Thank you, Nick. Great job.
Nick, that was amazing. I don't have a question so much about your research, but for your slide where you talked about the 20 things that you learned from your experience, would you be able to share that with us? Because I think some of my undergrads should really see that.
Yeah, absolutely. I would feel free. I would be more than happy to send that. And I can make it in more of a, that was like a very informal list. I just thought about that in like 10 minutes. I bet I could investigate to see what else I learned. But yeah, I would love to share that with anybody who would want it, because again, research, like it should be as a mode for teaching at a teaching institution, in my opinion.
Yeah, thank you.
If anyone has questions, feel free to speak up.
You don't have to be easy. I love some tough questions.
Well, I guess, I have a question. So you found that it's not the canopy openness that determines where microstegium is at LCW, but because it seems to be pretty common in gaps from tree falls and also in streams, would you say that it's just disturbance?
Yeah, honestly, it's probably disturbance. And in old growth forest, tree fall gaps are kind of one of the only forms of disturbance that occur pretty readily. Of course, there could be landslides, but that's not too frequent. And then also forest fires as well could be a form of disturbance. There are a myriad of types, but that is just the most common in LCW specifically. So I would say so. And then also, usually along these forests in the streams specifically, there are animals, of course, that could spread the seed as well. So that is a form of disturbance for helping the spread of the species.
Thank you.
Hey, Nick, I'll throw you a, sorry, Pat, where you can go ahead.
You're fine,
Nick, since you asked for challenging questions, I'll throw one out about statistics. And so you use correlation to kind of compare openness and biomass and openness and percent cover in this kind of series of steps of individual analyses, correlations. So are you familiar, or have you thought about like other ways that you might kind of combine all those to look at potentially how various variables might be like interacting with each other, or how responses might even be related to one another?
So Dr. Koslow and I have talked a few times, extensively, I would say almost, about the statistics. And what I've personally seen is biomass and percent coverage are measuring the same exact thing. So really we only have those two variables. So there wouldn't be any way to see if there's an interaction among the three. So I would say that from our statistics that it would be kind of misguided to do a correlation among the three.
Well, there are ways to even combine those response variables and to synthesize them into a single variable that might reflect some subtle characteristics about them that might not stand out on their own, like ordination type methods. So I'm not saying you should be doing that. I just wanted to see if you knew anything about that.
Yeah, I don't. I haven't taken BIOS yet. Yeah.
Nick, I have two questions, kind of more global questions. So if you could repeat the efforts, your particular research program from past summer, what would you change in terms of your approach, if anything?
Oh, that's a phenomenal question. I bet I would say that I would want to add more variables in the field that we are analyzing. Most of the projects, some of them measured pH, which we did think about, but then there was another intern who was doing pH, so I decided not to. So I don't know about pH specifically, but some people did the soil composition, because I mean, plants grow in the ground, and I think that that's often a neglected factor in plant ecology. Is the soil is so just not understood in science, in my opinion. There's so much rich diversity and interactions in the soil that are just so hard to understand because we can't see it. And that's what it comes down to, is things we can see are so much easier to understand. So
Right.
I wish we could have looked at the soil more, but again, it was tough because it was a short program. So it's hard to jam pack a lot of things in there, because we designed and put together a whole entire research project in 3 months with an output and a short article that went to the public afterward, as Dr. Brown and Richter can talk about. But yeah, I would say that that's what I wish I could change, but I don't know if I'd want to add that much pressure onto the program in itself.
And then a second question. So you mentioned, citizen science, or rather communicating with the public what we do. And you already made some very cogent points about the value of research in terms of teaching. And so if you could give an elevator talk, 30-second chat to a particular individual who is not that familiar with our institution, what was the biggest benefit you obtained then from this particular experience as an undergraduate?
Yeah, so the biggest thing I got from this experience was coming back to the learning outcome of the experimental design. There is very few opportunities for undergraduates to completely do their own project from start to end, which is not a problem. I think that going along with another project a professor has at an institution is great, because then you're adding into a bigger question that's being asked. But sometimes asking your own small questions really teaches you a nuance that would not be obtainable until you're a professor. So I think that that is a privilege that I got to experience, was working with Dr. Koslow's close mentorship and Dr. Watson and Robert to really go from start to end, which is a fascinating experience, because if I want to do this in the future in grad school, at least I know what path I need to take. It really turned on the lights for all the paths I need to see from start to end. So now I know where I'm going for these bigger questions I'll be asking.
Great, thank you, Nick, great.
Also, I wanted to add, along with like the connection to the popular and just going back to the public, is like a lot of the times when I'm looking at professors that I want to study under in grad school, I often look at their CVs to see if they have a popular articles or if they write op-eds, because it's so important to me that like if they're not communicating back to the public or training other undergrads or graduate students, then I'm often not interested, because that's just, I think it's being super ignorant and just thinking science is so uppity and you don't have to go back to the public level. But I think that the public is of the utmost priority when it comes to science.
Nick, it is both in terms of the science itself, say forest ecology, for example, plus also a matter of self-defense. We now have science under attack from a number of quarters, both infectious disease as well as climate change and things like that. So you're pointing out that the current generation of scientists and future generations need to be focusing, as you say, on addressing the public. NIH does a great job of this. Every year, Francis Collins goes before Congress and says, your money helps us do these things, so please fund us. So they're very good at NIH of actually publicizing to the representatives in Congress how their research dollars are spent. So I think it's a very good point to raise here that you're doing, that we should all be involved in, as Stephen and Kelly and David are, and others on our faculty, Jen and other people, communicate to the folks out there why it is so important we do our particular science and how that actually benefits them and helps us get to a better world ideally. So very good point, Nick. Thank you.
Hey, Nick. Awesome talk. I just had a follow-up to that just to be careful about using publications in the public, op-eds and so on, to judge that, because a lot of faculty don't write those. We are really active with obviously teaching, but outreach, going to schools, social media, and other venues that aren't published. So I don't know that many of us actually write for the public. And based on at least the most recent years, writing for the public may not be as important as social media, quick snippets.
Yeah, that's a good point. I did make an overgeneralization, maybe just more generally of like interacting with the public and having that reflected on doing internships like the Division of Natural Areas does heavily and interacting with the public. So I'd say it's more of an interaction, an intentional interaction with a citizen, because if you can explain a super complex concept in science to a fifth grader, then you're doing good in my opinion.
Nick, I have a quick question. So to kind of piggyback on education for all. So if all of us here were to come in contact with this and decide we wanted to remove it, what would be the best practices of doing that? So, you know, what time of year does it typically seed? And like, if we're pulling it, you know, if there are any roots left over, is that likely to regrow? Like what?
There is, I just dropped in the chat the Kentucky Native Plant Society article that I wrote up. It
Oh,
cites
perfect. Sorry.
specific research articles, but it depends on the size of the population. If you feel like you can pull it, do that, because then you don't have to get chemicals, which I'm kind of against when you have small populations, so that you can really make sure you don't change the soil chemistry. If it's too big, then of course going the mechanical route is good, but the time of year is super important when it comes to microstegium. Right now, it's almost a bit too late to pull it, but you do want to pull it in August, September, because too much before, then it can regrow to make sure there's flowers, but too late, then it's already seeded out.
Thank you. An awesome presentation, by the way.
Thank you.
Any other questions for Nick? Suppose not. Well, thank you very much again, Nick, for giving your talk. It was a good one.
Yeah, thank you all
And
so much for listening.